This book is about molecular virology. That is the molecular basis of how viruses work. It looks at the protein nucleic acid, and protein lipid interactions which control the structure of viruses particles, the ways viruses infect cells, and how viruses replicate themselves. Later, we will also examine the consequence of virus infection for host organisms. But it is important to consider the basic nature of viruses first. To understand how our present knowledge of viruses was achieved, it will be useful to know a little about the history of virology. This helps to explain how we think about viruses and what the current and future concerns of virologists are. There is more biological diversity between different viruses than in all the rest of the bacterial, plant, and animal kingdoms put together. This is the result of the success of viruses in parasitizing all known groups of living organisms, and understanding this diversity is the key to comprehend the interactions of viruses with their hosts. The principles behind some of the experiment techniques mentioned in this chapter may not be well known to our readers. That is why it may be helpful to you to use the further reading at the end of this chapter to become more familiar with these methods, or you will not be able to understand the current research literature you read. In this and the subsequent chapters, terms in the text in bold red print are defined in the glossary at the end of the book. What are viruses? Viruses are submicroscope obligate intracellular parasites. Most are too small to be seen by optical microscopes, and they have no choice but to replicate inside host cells. The simple but useful definition goes a long way toward describing viruses and differentiates them from all other types of organisms. However, this short definition is not completely adequate. It is not a problem to differentiate viruses from multicellular organisms such as plants and animals. Even with the broad scope of microbiology, covering prokaryote organisms as well as microscopic eukaryotes such as algae, protozoa, and fungi. In most cases, this simple definition is enough. A few groups of prokaryote organisms also have specialist intracellular parasite life cycles and overlap with this description. These are the Ricci and Chlamydia obligate intracellular parasitic bacteria, which have evolved to be so cell associated that they can exist outside the cells of their host for only a short period of time before losing viability. A common mistake is to say that viruses are smaller than bacteria. While this is true in most cases, size alone does not distinguish them. The largest virus known, current Pythovirus ibericum, is 1,200 nanometers long, while the smallest bacteria, A.g. Mycoplasma, are only 200,300 nanometers long. Nor Nor does genetic complexity separate viruses from other organisms. The largest virus genome, Pandora virus, 2.8 mbp million base pairs, approximately 2,500 genes. It's 20 times as big as smallest bacterial genome, Tremblia principes at 139 kbp, 1000 base pairs, and with only 120 20 protein coding genes, although it is still shorter than the smallest eukaryote genome.